This video is for the ELISA lab. ELISA is an acronym that stands for Enzyme Linked Immunoabsorbent Assay. The purpose of our lab is going to be to test to see if a person has antibodies against a specific disease. If a person has antibodies, that means that their body has identified a foreign pathogen and has started an immune response against that pathogen. A person who is not infected would not have their immune system fighting, and thus they would not have the antibodies. So by determining, are there antibodies, we can decide, yes, there are antibodies, and thus this person is infected, or no, there are no antibodies, and thus this is a healthy person. To do this lab, we will use a 96 well plate like we have used in several of our other labs. We don't need all 96 wells, so we are using just the top 2 rows. The first thing that we're going to do in this lab is in all of those wells in the top 2 rows, so these 24 wells here. In each of those 24 wells, we are going to put a squirt of antigen. An antigen is a piece of a pathogen. So we're putting a little piece of the pathogen into all of those wells. Secondly, we are going to add a squirt of blood into each of those wells. The blood may contain antibodies. An antibody is a protein found in the blood that targets a specific pathogen. If the person has the antibody will be attracted and bind to the antigen, which is what we see here in the second picture. The patient's blood had the antibody, so the antibody sticks to the antigen. If the patient's blood has no antibody, then there would be no binding occurring in that well. Thirdly, we will put a third squirt, and the third squirt will contain a man-made antibody that can change color. That man-made antibody attaches to the blood antibodies. So essentially, if we see a color change, that will tell us that the person has antibodies and thus the person is infected. If we see no color change, that would tell us the person does not have antibodies and thus the person is healthy. In my well plate, I have a few things going on. The first 3 wells represent a positive control. The purpose of a positive control is for me just to make sure that all of my chemicals and all of my procedure are working properly. In the positive control wells, I should see a color change. I do. I see purple. That tells me my chemicals are good and my procedure is good. The next 3 wells all represent a negative control. The purpose of a negative control is the same. In my negative control, I should see no color change, again indicating to me that all of my chemicals are working and my procedure is accurate. So those first 2 sets of 3 help me just to make sure that everything is going properly. After that, I have several patients. I have 3 wells that represent patient A. I have 3 wells that represent patient B, 3 that represent patient C, 3 that represent patient D, 3 that represent patient E, and 3 that represent patient F. In each of the 3 wells for each patient, the sample was tested in triplicate, meaning in 3 times. So you can think about, why would we test something more than once if we're looking to determine if a person is infected or healthy? So for example, these F wells all are the same patient, patient F. We just did 3 identical tests on them. From this picture that's given to you, you can determine if the patient is healthy or not. A healthy patient would have no antibodies, and thus the wells would remain clear. An infected person has antibodies, would have a color change, and they would display as purple. So you're going to need to make a judgment call on if you see purple or not. I would say any purple is purple infected. And then only if you see totally clear would you then say that they're healthy.